Welcome to another lecture by MedicoMedics, Learning Made Easy, Genetics Chapter 3, Molecular Mechanisms of Inheritance. In this lecture, we will repeat and give you an overview over DNA replication, transcription, translation, and end with a summary. Now, DNA contains the instructions for building and maintaining an organism. Key processes include replication. This is where DNA is copying itself before cell division. Another one is transcription. This is where DNA is converting to RNA, more specifically, messenger RNA. Then we have translation. This is where the RNA is used to make proteins. And regulation ensures only the required genes are active. So DNA is like a library of instructions, and replication makes a backup copy, while transcription writes the notes. And translation then uses these notes to make proteins. Now, DNA replication, basics. So what happens here? Well, DNA makes a copy of itself, and this occurs in the S phase of the cell cycle. And this is an illustration of the cell cycle. And the synthesis phase is illustrated here in green. Now, the key enzymes involved are helicase, and this enzyme unwinds the DNA helix. We then have DNA polymerase. This adds new bases to the growing strand. And then we have ligase, which seals gaps in the DNA backbone. The outcome? Two identical DNA molecules, each with one old strand and one new strand, also called semi-conservative replication. Now, the steps in detail. So we have the terms initiation, elongation, and termination. So during initiation, helicase opens the double helix at replication origins. During elongation, DNA polymerase adds complementary bases, AT, so adenine and thymine, CG, cytosine and guanine. And then towards termination, ligase seals the DNA, completing the process. Now, mutations can occur if proofreading mechanisms fail. For example, an error in replication can lead to conditions like sickle cell anemia if not corrected. So DNA makes a near-perfect copy of itself, but mistakes can happen, and this sometimes lead to genetic diseases. Now let's repeat the process of transcription, where we are converting DNA to RNA. So what happens? DNA is transcribed into messenger RNA, and this takes place in the nucleus. Key players here are RNA polymerase, which copies DNA into RNA. We then have promoter regions, so the DNA region signaling where transcription begins. So to the question, where does transcription begin? It is in the region called the promoter region. Then we have two other important concepts like intron and exons. So non-coding are introns and coding regions are called exons. The important steps included initiation, where RNA polymerase binds to this promoter region. Then we have elongation, where RNA polymerase synthesizes RNA. And then we have termination, where RNA polymerase releases the RNA strand. And a way to remember is that transcription is like writing down instructions from a book, so the DNA, onto a notepad, which would be the RNA. And then this RNA can leave the nucleus and then through the process of translation, start producing proteins. Now then, translation, the making of proteins. So what happens? Messenger RNA is translated into a protein, and this takes place in the cytoplasm at ribosomes. Key players here include messenger RNA, so it's the messenger with the instructions. We have transfer RNA, which brings amino acids to the ribosomes. If you look at the illustration here, you can see we have the messenger RNA. So its primary structure. We have the ribosome itself. We have the transfer RNA bringing amino acids. And then the machinery then assembles this to make proteins. Steps here include, again, initiation. So ribosome binds to the messenger RNA. These two. Elongation. So transfer RNA delivers this amino acid, forming a chain. And then termination. A stop codon signals the end. When it's done, it signals stop, and the protein is then released. So translation is like using a recipe, mRNA, to assemble a meal, which would then be the proteins. And these proteins are the building blocks of your body. Now that you've come this far, indeed, there are a lot of new terms, a lot of different steps, and it's easy to be confused. So we will provide a mid-summary here in this lecture to try and contextualize for you all the different parts that we just went through. Now we have something called the central dogma of molecular biology. So ideally going from DNA to proteins. Now the process of genetic expression begins with DNA replication. It's ensuring that genetic material is faithfully copied before cell division. Okay, so we have DNA replication to begin with. And this starts with the enzyme helicase, if you remember from our previous slides. And this enzyme is responsible for unwinding this double helix, creating something called a replication fork. Then we have DNA polymerase. Okay, it's not RNA polymerase, that comes later. DNA polymerase adds a complementary nucleotides to each of these original strand, synthesizing the new DNA. Now, since DNA strands run in opposite directions, replication occurs differently on each. The leading strand is synthesized continuously, while the lagging strand is made in short Okazaki fragments. And these fragments are later joined by ligase, which seals the DNA backbone and completes replication. Okay, so we've had DNA replication happening. Check. Now, once the DNA is replicated, gene expression begins with transcription, where a gene's DNA sequence is now copied into messenger RNA. All right? After that, RNA polymerase, not DNA polymerase, but RNA polymerase, binds to the promoter, a specific DNA sequence signaling the start of transcription. So it binds to this specific place, and that's the kickoff of transcription. Now, this marks the initiation phase, right? Phase 1, initiation. Now, after that, we had step 2, which was elongation. So RNA polymerase moves along the DNA, adding complementary RNA nucleotides. So adenine, uracil, cytosine, and guanine. If you remember, in DNA, we had the pairing of adenine to thymine and cytosine with guanine. But when we're talking about RNA, we have the pairing of adenine with uracil and cytosine with guanine. Now, once this reaches a termination signal, then transcription ends. And the newly formed pre-messenger RNA undergoes processing. Now, this includes removing introns, which are non-coding regions, and splicing together exons, which are coding sequences, forming the final mRNA. All right? Now, this processed mRNA now leaves the nucleus and enters into the cytoplasm. And this is where translation starts. So up till this point, we were in the nucleus of the cell. Now we're in the cytoplasm. And in the cytoplasm, where translation starts, this is where the RNA message, right, from the messenger RNA is now translated or converted into a protein. And this process occurs in the ribosomes. This is the organelle, the ribosome, which reads the mRNA sequence in 3-letter codons. So during initiation, a transfer RNA, tRNA, carrying methionine, or methionine, I think it's called, binds to the ribosome at the start codon. And this start codon is abbreviated AUG. During elongation, more transfer RNAs bring amino acids, matching their anticodons to the mRNA codons, while the ribosome links the amino acids together to form a growing polypeptide chain, so a chain of amino acids. And then finally, in termination, the ribosome encounters a stop codon, promoting the release of this completed protein. So this is a step-by-step journey from DNA replication to transcription, and finally to translation, ensuring that genetic information is accurately preserved and used to build functional proteins which drive all cellular activities. Hope the clarification was clear. We also go into depth in our cell biology course for those who are interested to check that out. So in summary then, DNA replication ensures genetic material is copied. Transcription converts DNA to RNA, and translation turns RNA into proteins. Gene regulation controls when and where genes are active. Errors in these processes can lead to genetic disease or variations. And that's the end of Lecture 3. Continue now to Lecture 4.